So now that we've learned about associative learning, in other words, classical and operant conditioning, we're going to turn our attention to non-associative learning very briefly. So we're going to, hopefully by the end of the video, you'll understand how non-associative learning is different from associative learning. And there's also an extra video linked in the module pages that will help you understand as well. And we also want to understand what's happening at the synapse during this type of learning, because what we'll see when we go in subsequent videos, when we start talking about long-term potentiation, is that there are some similarities between what's happening in non-associative learning at the synapse and what we're going to see in long-term potentiation. So while it may not seem intuitive, there is a lot of studies on learning that looks at what's happening in the brain that's done in invertebrates, in particular the aplysia. Now, there is an extra video linked on the module pages that you should watch, because it will tell you a little bit about aplysia, give you a little bit more insight into what we're talking about. But it is used often as an experimental model, and it is a slug-like invertebrate. So I actually was super excited, probably, maybe 7-8 years ago now. My kids and I went down to the tide pools down near San Pedro by the Cabrillo Aquarium, I think it is. And we're out there and we're looking, we're trying to see if we can see anything cool. And all of a sudden my daughter goes, "Mom, what's this?" And I was like, "Carrie, that's an aplysia." And then we went inside to the aquarium and we saw it. And so I for me, being, coming from neuroscience, I just thought it was the coolest thing to actually see one in person. I never actually did research on them. But they were studied because they have very large neurons. And the type of learning that's studied in them is called non-associative learning. The idea, it's non-associative because the animal's learning something, but they're not making association between two different stimuli like we saw in classical and operant conditioning. So the types of learning that are studied in an aplysia are going to be habituation and sensitization. So habituation is where you have a decrease in a response to a stimulus that's presented repeatedly. So if you think about an aplysia, it's a sea snail or a sea slug, right? And it sits off the coast, typically in colder waters in the, Northern California, I think. But you're going to have these, in the tide pools, you have these waves. When the tide is low, at least, you have these waves lapping up against you all the time. And as you saw in the video, when the siphon is touched, the gill is going to come in, right? And so the animal habituates to these types of things because it doesn't want to have to waste all that energy every time a wave comes up to bring in that gill, right? So it's a decrease in response to a stimulus that's repeatedly presented to it. So again, the idea is that that the gill will withdraw, but that response will decrease over time. And so what we see here in a control animal, this is you stimulate the siphon, you see that withdrawal, and you see this is normal neurotransmitter release. In an animal that's habituated, you can see that there's a decrease in the amount of release. It's not a decrease in the number of connections, but a decrease in the neurotransmitter release. Now, sensitization, on the other hand, is where there's an increase in response to the mild stimulus as a result of previous exposure to more intense stimuli. And there's a video on the module page that shows that sensitization. And the idea is that now you're being it's more sensitive to a stimulus. And so there's more release of neurotransmitter at those synapses. So that, in short, is a brief description of non-associative learning, how we use aplysia to study that type of learning, and all that we've learned from the aplysia. Again, there's changes happening at the synapse with decreases in neurotransmitter, increases in neurotransmitter. We'll see with LTP changes in the synapse size, number of receptors, all those kinds of things happen.